Hello, good evening. How are you all? Am I audible, everyone? Can you all hear me? Hello? All right. So, welcome to this session 3, everybody of Prarambh. Okay. All right. Good evening, everybody. All right. How are you all doing? Okay, so today we are going to do our session 3, and today we are going to discuss about chromatin structure and function. See, you all are from biology field, right? So you all know that we have cell, that is the basic unit of life. Cell has nucleus. Inside the nucleus the chromosomes are present, and the chromosomes are the one that contains the genetic material, the blueprint of how a cell should work, that is stored in the DNA, right? Now I'll tell you an astonishing fact that, okay, can somebody tell me how many chromosomes are present in human cell quickly? Yes, anybody? Yeah, 46. Correct. 46 chromosomes, right? But what is this? 46 chromosomes, that is the diploid number, right? So you say 46 or you say 23 pairs of chromosomes are present, right? So what is haploid? Absolutely correct, Jason, Shramalina. Yeah. So people who are saying 46 is also correct. If you are saying 23 pairs, that is also correct, right? Now, this 23 pairs of chromosome that is present, which is called as the diploid number, right? That is, each chromosome is present in two copies. Now, do you know, if I join the DNA of each of these 46 chromosomes end to end, okay, do you know how much long it will be? Any idea? Some wild guesses if you want to make? If you join the end-to-end DNA of this 46 chromosomes from a single human cell, how long will it be? Yes? Any idea? Any guesses? Anybody? Absolutely correct, Nishant. Yes, it will be approximately 2 meters long. And how big is our nucleus? It is merely in micrometers. So, can you imagine how this 2 meter long DNA can be packed inside a structure which is merely micrometers wide? It's astonishing, right? That is exactly what we are going to study today. Okay, how those DNA, how those long DNA are coiled, recoiled, supercoiled. Okay, and that's how it will be packed inside the nucleus of a cell. Okay, so yes, exactly, Shivani. So that is what we are going to discuss. All right, so we are going to discuss everything about chromatin structure and function. Okay, and not only in eukaryotic cell, we are going to talk about in viruses, in prokaryotic cells, as well as in the eukaryotic cell. So welcome to the session, everyone. All right, so if I talk to you people in Hindi in between, will you people be comfortable with that? Just let me know. Okay, Jason says not at all. Okay, all right. Okay, yeah, most of the people are saying yes. Okay, so what I will do is I will try to repeat something in Hindi. Okay, and if you guys also are not able to understand anything in English, please don't hesitate and ask me to repeat in Hindi. I will do that. Okay, all right, let's start then. So, what is the organization of DNA in the chromosome? So, if you see a genome, it is the full amount of genetic material which is found in virus, a prokaryotic cell, or even an eukaryotic cell. Okay. So, genetic material is present in all, in viruses, in prokaryotic cell, as well as eukaryotic cell. In viruses, the genome can be DNA or RNA. Okay. Or it can be found in one or more pieces. Okay. In prokaryotes, the genome is usually, but not always, a single circular DNA. Okay. All of you know, right? So if a prokaryotic cell, it has a DNA like this, right? A circular, covalently closed circular DNA, which we also called as the cccDNA. Now, in case of eukaryotes, the main genome of eukaryotes is typically distributed among the haploid set of chromosomes in the cell nucleus. Okay, now this eukaryotic cells, along with the nucleus, they can also have some genetic material in some of their organelles as well. What are the organelles that have their own genome? Yes, can anyone tell me what are the eukaryotic organelles with their own genome? Yes, Shivani, mitochondria, correct. Yes, Vishal. Yes, anything else? Any other organelle do you know? Chloroplast, absolutely correct, Jason. So, mitochondria and chloroplast, they are the two organelles of eukaryotic cells that have got their own genome, right? So, apart from this organelles, okay, so there will be majority of the genetic material in the eukaryotic cells are packaged inside the nucleus, okay, and the type of genetic material that they have is usually linear, okay, it is in the linear DNA form, right? Now, once the scientists they understood that the DNA is the genetic material, definitely the next question to answer was how that genetic material was organized within the genome. Okay, so if you see the organization level, so it is actually DNA which comes in association with various kind of proteins that makes it a chromatin. Okay, and this chromatin is further condensed and further packaged, and that is when it forms the chromosome. Okay, I have often seen people getting confused between these terms. So DNA exactly is not chromatin. DNA plus protein forms the chromatin, and chromatin after packaging, okay, then it forms the chromosome, further condensation. Okay, so, DNA organization structures have been studied using numerous approaches, including biochemical analysis as well as visualization by light microscopy and electron microscopy. So, understanding the organization of DNA is also important to clearly understand the process by which the information in a gene is accessed and processed. Okay, so we will be discussing all these things, and I am sure at the end of this session, you will have a clear understanding of how DNA is packaged into chromatin, how it is assessed at the time of time of transcription, and all those processes. And as usual, at the end of the session, we will discuss some of the previous year questions as well. And as you people know, stay connected. And at the end, you will get the notes of this session. Okay, as you fill up the feedback form, you guys will receive the notes of the session. Okay, so आप सब लोग बने रहिए एंड तक सेशन के. जैसे ही आप लोग फीडबैक फॉर्म भरेंगे, आपको नोट्स मिल जाएगी इस लेक्चर के. Okay, so we will start with the viruses. Okay, so we'll start our discussion from viruses, then prokaryotes, and then finally we will move to eukaryotes. So viral chromosomes. Now, depending on the virus, the genetic material may be double-stranded DNA, single-stranded DNA, double-stranded RNA, or even single-stranded RNA. Okay. Now, viruses are the only cells, okay, or organisms that will have DNA or RNA as their genetic material. Okay. Otherwise, if you talk about prokaryotes or eukaryotes, they will only have DNA as their genetic material. Now, in the viruses, this DNA and RNA, it can be the genetic material, whatever is there, is can be circular or linear. The genomes of some viruses are organized into single chromosome, whereas other viruses have a segmented genome. Okay. Now, what do we mean by segmented is that the genome is distributed among a number of DNA molecules. Okay. Just like human cell, if we see, our DNA is also segmented. Okay. Because we have multiple chromosomes, right? Our DNA is divided into multiple chromosomes, right? So in the same way, viruses also, some of the viruses can have a single chromosome, or they can have a segmented genome where the DNA is whatever is the genetic material is segmented into multiple molecules. Okay. Now, examples of viruses with DNA genome. Okay. Now, yes, questions can be asked, direct question can be asked about the genetic material of the viruses. Okay. So I have given here some examples. So T2 bacteriophages, or the T series, okay, T even bacteriophages, they are all having DNA as their double-stranded DNA as their genome. Then herpes virus and Gemini virus, that also has double-stranded DNA as their genome. Okay. Now, 5X174 and parvoviruses, these are examples of single-stranded DNA genome, okay, having single-stranded DNA as their genome. Now, what are the viruses with RNA genome? There is reovirus, which has double-stranded RNA genome. And apart from that, poliovirus, influenza virus, SARS-CoV-2, the one which caused corona outbreak in the world, okay, for last 2 years, that is also a single-stranded RNA genome. Okay, everything followed so far, everybody? Let's have some responses, everyone. All right. So, how this genetic material is packaged in the viral genome? Okay. Now, this is the usual structure. If you see, so viruses, they will have an envelope. On the envelope, there will be the spikes. Okay. And inside the envelope, there will be the capsid protein. And in the capsid protein, whatever is the genetic material, that will be packaged. Okay. So, whether it's RNA or DNA, whatever is the genetic material, it will be packaged inside the capsid. So, viral nucleic acid molecules have been visualized with the electron microscope. Okay. And you know what has been found? That one constant feature is shared by viruses, bacteria, and eukaryotic cells. And that constant feature is the ability to package an exceeding long DNA molecule into a relatively small volume. ओके सो यही चीज हर ऑर्गेनिज्म में देखी गई है कि वायरस हो कि प्रोकैरियोट्स हो बैक्टीरिया हो या यूकैरियोट हो एक बड़े से डीएनए मॉलिक्यूल को बहुत अच्छे से पैक करके एक बहुत छोटे स्ट्रक्चर के अंदर पाया जाता है okay it's not only in viruses in prokaryotes in eukaryotes this feature has been seen constant okay so if you just see the lambda phage the DNA is 17 long and must fit into a phage head which is less than 0.1 on one side on any side okay so you can see the kind of compaction the level of compaction that is required here. In many cases, almost all space is filled, indicating nearly perfect packing. Once packed within the head, the virus's genetic material is functionally inert until it is released into the host cell. Okay, that is the uniqueness of about virus, right? So it kind of lies in the threshold of living and non-living, that when the virus all is living when inside the host because it is replicating, but outside the host it cannot replicate. Okay, so the genetic material is all encapsulated inside the capsid, inside the envelope, and it stays inert. But when it attaches with a host, then it injects its genetic material inside the host cell. Okay, and then this genetic material hijacks the host cell machinery and replicates. Now, before going ahead, yes, if you want to get more classes, podcasts, webinars, courses, everything is available in BioTecNika app, which is available in Apple Store as well as Google Play. Please download it from here there, and please join our BioTecNika Telegram group as well. The link of it is shown here. All right. Next, moving on to prokaryotic chromosomes. Now, most prokaryote contain a single double-stranded circular DNA molecule. Okay, so it's almost DNA and it will always be double-stranded and circular. Most of the prokaryotic cells, they will have a single double-stranded circular DNA molecule. Okay, so if you see the prokaryotic structure, okay, so I hope all of you know this, right? So this is the prokaryotic structure. So inside the cytoplasm, prokaryotes, they do not have any membrane bound organelles, right? So whatever is present in the cytoplasm directly. So DNA is not enclosed in the nuclear membrane that we see in eukaryotes, but in prokaryotes, that it is present in a condensed region called as nucleoid. Okay. Now, most of the prokaryotes, they will have only one DNA, one circular double-stranded DNA as their genetic material. Some other prokaryotes can also have, apart from that one circular double-stranded DNA, can also have some more chromosomes, which can be present as circular or linear form. Okay. So usually what happens is they will have one main chromosome. Okay. This one, which is called as the genetic material of the cell, and along with that it will have some plasmid DNA. Okay, and this plasmid DNA is also circular, is also double-stranded DNA molecule only, which has the ability to autonomously replicate. Okay, that is, its replication is not dependent on the cell's replication. It can replicate on its own. Okay, and it may or may not be essential for the life of the cell. Sometimes these plasmids, they contain the genes that provide the cell with the ability to survive or resist certain antibiotics. Now, in bacteria and archaea, the chromosome is arranged in a dense clump in a region of the cell known as the nucleoid. So, E. coli DNA approximately is around 1100 micrometer long. Okay, this E. coli DNA, if you see, is around 1100 micrometer long, which is packaged inside the cell, which is around 1000 times smaller in length. Okay, so how much packaging it has to done? So the DNA fits into the nucleoid region of the cell in part because it is super coiled. Okay, that is, the double helix is twisted in space about its own axis, and that is what forms the supercoiled DNA. Okay, now just to give you an idea about how much supercoiling will condense the structure, so you can see this is the electron microscopy. This is how the DNA will look like from a bacterial cell, a relaxed DNA, relaxed double-stranded circular DNA. Okay, and this is the same DNA when it has been supercoiled. Okay, so you can see how much less space it is occurring when it is supercoiled. And you can also imagine it with the rubber band. Have you people ever twisted the rubber band in hand? We often fidget with it, isn't it? Anybody can relate to it? Have you people twisted a rubber band? Or if you have a rubber band handy, you can try it right now. Yes, anyone? All the attendees, I would want some responses, which I'm not getting. Let's make this session interactive. Come on. All right, Nishan. So you have done it, right? You have twisted a rubber band, and do you see that when you coil it and supercoil it, how much small it becomes? What about others? Anybody? No one else? Okay, Manisha. All right. Okay, now I'm getting some responses. Yes, keep the responses coming, guys. Let's make this interactive. Okay. So, for those people who don't understand supercoiling, so to understand supercoiling, consider a linear piece of DNA with 20 helical turns. Okay, so this is a linear DNA with 20 helical turns. Okay, that means 20 times one strand is rotating over the other. Okay, it then what you do, you simply take the ends here, this two ends and this two ends, and join them. Okay, so if we simply join the two ends, we have produced a circular DNA molecule that is relaxed. Okay, now if instead we first untwist one end of the linear DNA molecule by two turns. Okay, so what we did here, see, we opened two turns, right? And then we joined the ends, then we took this two and joined it here. Okay, and then join the ends, the circular DNA molecule produced now will have 18 helical turns, because initially it had 20, but then we opened two, right? So it now has 18 helical turns, okay? And a small unwound region, a small region which is open, okay? But this structure is not energetically favored, okay? It is not in a relaxed state, it has a tension in it. Okay, so if you keep a structure like this, so what this structure will do is immediately it will switch to a structure with 20 helical turns and 18 super helical turns. Okay, what will be super helical turns? Like this, where both the helix, okay, will again twist among each other, around each other. Okay, and that is what is called as supercoiled form of DNA. Do you people get the idea what is supercoiling? Okay. Now, when we say to supercoiling, yeah, Shubham, see here, this two bands. Okay, so here it is just two strands of DNA. They are twisting around each other. Okay, that is the normal helix that we have, right? Now, when two different strands, okay, see here, this is two strands and another two strands. Okay, when they also coil among each other, around each other, that is what is called as supercoiling. Okay, so now there are two types of supercoiling: negative supercoiling and positive supercoiling. Okay, now you don't have to get confused with the positive and negative here. Okay, this positive and negative terms that are simply given to they simply refer to their relative direction with the helical turns. Okay, so if you have a bDNA, what side turn it has? The helical turn in the DNA is which side? Anybody? Which side is the helical turn in DNA? The usual DNA that we mostly find in all the cells. Right handed, correct. Okay. So if you do a supercoiling again in the right hand side only, okay, that will introduce positive supercoiling. All right. And if you do a supercoiling in the left hand side, then it will called as negative supercoiling. Is it clear? Okay, because the DNA helix is turned is present in a right-handed turn. Okay, so if supercoiling is done right-handedly, it will be called as positive supercoiling. If the supercoiling is done left-handed direction, it will be called as negative supercoiling. Okay, got it, everybody? Right? But in terms of condensation, if it is matter, or in terms of compactness of the DNA, if it matters, it does not matter whether the DNA is positive or negative supercoiled. Okay, anyhow, if their supercoiling is there, it will cause the DNA to become more compact. ओके, इससे फर्क नहीं पड़ता है कि डीएनए किस साइड में कॉइल्ड है, सुपर कॉइल्ड है, ठीक है, अगर वो पॉजिटिव भी है या नेगेटिव भी है, दोनों डायरेक्शन में ही it will just compact the DNA. Okay, so don't think like if it is positive supercoiling then it is compact, and if it is negative supercoiling then it will become relaxed. No, whichever supercoiling it is, positive or negative, it will result into compaction. It will make the DNA more compact. Now, the amount and type of DNA supercoiling is controlled by an enzyme which is called as topoisomerases. Okay, the enzyme that are found in all organisms. Okay, so this topoisomerase is found in prokaryotes as well as eukaryotes. All right, and this topoisomerase is responsible for maintaining the DNA in supercoiled form. Okay, so just have a little bit idea about what is topoisomerase. So there can be two types of topoisomerase, type 1 and type 2. And that will depend on whether they cleave one or both strands in the helix respectively. Okay, so topoisomerase is responsible for supercoiling. All right, now if that supercoiling happens by cleaving one strand at a time, then it will be the work of topoisomerase type 1. Okay, type 1 will cleave one strand at a time. But if it is type 2, then it will cleave two strands at a time to introduce more supercoiling. Okay, so there are two forms of topoisomerase. It's written here, type 1 and type 2. So type 1, it cleaves one strand at a time, and because of that, it increases the linking number by 1. Type 2, it cleaves both strand at a time, so it will change the linking number by 2. Okay, now type 2 topoisomerase, it requires ATP. Okay, and a subpart of type 2 topoisomerase is DNA gyrase, which is found in prokaryotic cell. Okay, so in this DNA gyrase is responsible for negative supercoils in the prokaryotes, and it's a member of topoisomerase 2. All right, so since it is a member of topoisomerase 2, so it will cleave two strands at a time, and it will require ATP. Okay, question regarding DNA gyrase has been asked previously. Yes, gyrase is present in prokaryotic cell. Correct, Manisha. In E. coli, topoisomerase 1 serves to reduce the number of negative supercoils in a closed circular DNA molecule. Topoisomerase 2 introduces negative supercoils in the DNA. Okay, so this is a relaxed DNA and this is a supercoiled DNA. Okay, so who did it? It will be the function of topoisomerase. Is it followed, everyone, so far? Yes, everybody, so far, everything clear? Can we move ahead? Come on, write some BIOs. Type 2. Shrivani, it's the topoisomerase 2 that requires ATP. Type 2 requires ATP. All right. Now moving on to the eukaryotic chromosome. So as you know, eukaryotic genome typically are distributed among several linear chromosomes because we have a segmented genome with the numbers characteristic to each species, right? It varies from different species to species how many chromosomes are present in a particular cell. Now, the total number of DNA in the haploid genome, DNA in the haploid genome of a species is what is called as the C value. Okay, and this C value will vary from species to species. Now, if you see on this side, I have given you the chart table that has the C value of different organisms. Okay, starting from viruses to bacteria to eukaryotic cell. Now, the C-value data that the amount of DNA found among organisms varies widely, and there may or may not be a significant variation among the amount between related organisms. Okay. For example, you see here these organisms. Okay, they are belonging to the eukaryotes, right? So all the higher organisms in eukaryotes, you see that they usually have they all have kind of similar the C value, similar number of C value, right? So C value will be kind of similar to between the related organism. Okay. Okay, Venkata, we are talking about chromatin structure and its function. So we have already seen what is the genetic material in viruses, how it is kind of packed. We also saw a little bit about the prokaryotes, and now we are moving to eukaryotic chromosomes. Shrivani, if the screen is not clear, please change the resolution. Okay. So we are talking about C value. Okay. Now this C value is the total amount of DNA in a haploid genome. Okay. Now it has been, so earlier the idea was that more complex the organism will be, more will be its C value, or the vice versa, that if an organism have higher C value means more DNA, so more will be the complexity. But after the C value of different species are obtained and compared, it was found that there is absolutely no direct relationship between the C value and the structural or organizational complexity of the organism. Okay, and that concept is what is called as C value paradox. Okay, for example, you see here you have amoeba whose C value is around 100 times more than that of humans. Okay, but complexity wise, see, amoeba is such a tiny organism, single cellular organism, whereas humans, we are so complex, right? So that is like, this is what it is, that there is no direct correlation between the C value of different species and the organizational complexity between them. Okay, and that is what is called as C value paradox. No, C means the total DNA content. C value is stands for the total DNA content of a haploid genome here. Total DNA of a haploid genome is what is called as C value. Okay. Now let's have a look of how DNA content of a cell changes during different stages of cell cycle. Okay. I'm very sure you all have some idea about cell cycle. Do you? Is there anybody who doesn't have any idea about cell cycle? Quickly, if you can let me know. Anyone who doesn't have any idea about cell cycle? थोड़ा बहुत सबको पता है, right? Cell cycle, even if it is not in detail, but थोड़ा बहुत सबको पता है, right? That there will be interphase. Interphase में 3 phases होंगे, G1, S and G2. and then there will be M phase, right? And apart from it, a diversion of the cell cycle will be a G0 phase, which is kind of a resting phase where the cells still stay alive, but they do not replicate, right? So how the chromosomes behave? All right. So eukaryotic cells, they reproduce in a cell cycle consisting of four phases: G1, S, G2, and M. Okay. And these three phases, G1, S, G2, are part of interphase. Okay. Whereas M phase is the division phase. Okay. So if the cell is undergoing mitosis, then that will also happen in the M phase. If the cell is undergoing meiosis, that will also happen in the M phase. Now, each eukaryotic chromosome is consists of one linear double-stranded DNA molecule running throughout its length. Okay, so one chromosome has one DNA. All right. So when you say that a eukaryotic a human cell has 46 chromosomes, that is the diploid number. So how many DNA molecules are present? Yes. How many DNA molecules? एक ह्यूमन सेल में 46 क्रोमोसोम्स होते हैं, है ना? तो फिर कितने डीएनए मॉलिक्यूल्स होंगे न्यूक्लियस में? चलो, टू बी मोर स्पेसिफिक, न्यूक्लियस के अंदर, एक ह्यूमन सेल के न्यूक्लियस के अंदर कितने डीएनए मॉलिक्यूल्स होंगे? Jason, it will be 46 only, no? Because one DNA, one chromosome. It won't be 46,000. It will be 46, right? Because if you have a single chromosome, if this is your chromosome, so in that chromosome running from top to end, there is only one DNA molecule going on. Do you get it, Jason? Okay, so each eukaryotic chromosome is consist of one linear double-stranded DNA molecule running throughout its length and complexed with about twice as much protein by weight as DNA. Okay, so I told you in the beginning itself that chromatin is not alone DNA. DNA plus proteins is what makes it chromatin, which on further compaction makes it chromosome. All right, but if you see number of DNA molecule, yes, there will be only one DNA, one chromosome. Okay, but it will be coupled with proteins as well. All right, so and in the S phase is called as the synthesis phase, right, where the DNA replication happens. Correct. So when the DNA replication happens, then what it will result? The entire DNA strand will be replicated, right? And that will result in the formation of sister chromatids. All right. So if you see the nucleotide sequence, so nucleotide sequence of the sister chromatids will be identical because they are basically formed from the other strand only, right? So after the S phase, now each chromosome has two sister chromatids, and each sister chromatid will have one DNA molecule from top to end. Am I clear with this, guys? So after DNA replication is done in the S phase, okay, now a chromosome will exist in a pair of sister chromatid, right? With one DNA molecule in each of the sister chromatid. Okay, but sister chromatids, they do not separate, like they separate from all other parts, but they keep joined through the centromere. Okay, and they will get separated completely only during the anaphase of cell division. Okay, so duplicated chromosome with two sister chromatids have one linear double-stranded DNA molecule running the length of each sister chromatid. Okay, so that is how they will be present, and only in the anaphase of the M stage, okay, they will separate. Okay, so at this stage where the sister chromatids are joined, so they remain joined, No, okay, Shrutika, you are asking a pair of chromosome and sister chromatids same. No. A pair of homologous chromosomes means they have the same set of genes present in them. Okay, so when we say that there are 46 chromosomes, that means there are 23 pairs of chromosome. Okay, that is different. But each chromosome, so there are 46, and each chromosome will replicate at the time of cell division, and that will form uh, each chromosome will have a pair of sister chromatids. Are you getting me, Shrutika? So a pair of chromosome will mean a pair of homologous chromosome which have the same gene loci present in them. Okay, but the alleles of the genes present may be same, may be different. But when you have sister chromatids, they are exactly replica of each other because they're produced by the replication, DNA replication process. And this sister chromatids, they will separate at the time of anaphase of the cell division process. Is it clear, everybody? Okay. Now moving on to the structure of chromatin. Chromatin is the so when we use some that chromatin comprises of DNA and proteins. All right. Histones and non-histones are two major types of proteins associated with the DNA in chromatin. No, Manisha, G0 phase, it can the cell can enter it after one cell division, okay? Or it's like, see, G0 phase is, it's not that all the cell has to go through that G0 phase, okay? So if a cell has to stop replicating, all right, then it enters this G0 phase where it remains viable, okay? It is completely viable, it is living, but it does not divide anymore. All right, like our nerve cells, the mature nerve cells, right, they don't divide, right? So they are in the G0 phase. All right, so yeah, Shivani, so I was saying that the chromatin that we said is the stainable material in the cell nucleus. Okay, so I hope all of you have seen the cell division slides in your graduation. Have you? You must have either prepared the slide on your own, or you must have seen the pre-prepared cell division slides. Yes or no? Right, so when you have seen a slide of cell cycle, okay, so in there what you see, we see only the chromosomes, right? Because this chromatin is the stainable material in the cell nucleus, okay, and this chromatin is made up of DNA plus proteins. Now what all proteins are present? There are two types of proteins that are usually present in the DNA. One is called as the histone protein, and whatever is not histone is called as non-histones. Okay, so histones and non-histones are two major types of proteins which are associated with the DNA in the chromatin. Okay, and this fundamental structure of chromatin is found to be identical in all eukaryotes. Okay, so right, so जो fundamental structure है, जो basic structure है chromatin का, आप किसी भी eukaryotic cell को उठा लो, उन सब में same है. Okay, so both of these proteins, histone and non-histone proteins, they play an important role in the physical structure of the chromosome. Okay, and that also plays a very important role in making the compaction. Okay, so that big 2-meter long DNA, okay, can be packaged within a nucleus which is just a few micrometer long. So let's have an idea about what are these histones and non-histones. So first of all, histones. Now they are the most abundant proteins in the chromatin. Okay, they are the most abundant. These histones, they are most abundant. Now these histones, they play a crucial role in the chromatin packaging. So without compacting, the DNA of the chromosome, as I was telling you, will be 2 meter long, right, of the entire diploid cell if we join it end to end. Okay, so because of that, if we need to go for so much compaction, that's why these proteins help. Okay, now this histones, they are small basic proteins, okay, proteins of basic nature, okay, with a like basic acidic wala basic, okay, not fundamental. All right, so they are small basic proteins with a net positive charge. All right, now DNA is negative charge, right, and these histones are positive charge. So we know that opposite charges attract, right? So because of that, so because these proteins are positively charged, they facilitate their binding to the negatively charged DNA. All right, now there are 5 main types of histones that are associated with the eukaryotic nucleus, eukaryotic nuclear DNA. They are H1, H2A, H2B, H3, and H4. Okay. Now the amino acid sequences of this histone proteins H2A, H2B, H3, H4 are have seen to be highly conserved. Okay, evolutionary speaking, and even between distantly related species. Okay, so what does this evolutionary conservation tells us? Any idea? सो व्हाट वी से इज दैट जब हम इसके अमाइनो एसिड सीक्वेंस देखते हैं, ठीक है, इन चारों टाइप के हिस्टोन्स में, तो वो आप किसी भी यूकैरियोटिक सेल में ले लो, एक बहुत सिंपल से सेल को ले लो या फिर बहुत कॉम्प्लेक्स से ऑर्गेनिज्म के सेल को ले लो, काइंड ऑफ इट हैज बीन फाउंड टू बी कांस्टेंट. Okay, so that means like they are found to be same or similar in the distantly related species. So that shows that it is evolutionary conserved. Right. So, evolutionary conservation, what does this tell us? So, it tells us that these histones, they perform the same fundamental role in organizing the DNA in the chromosomes of all eukaryotes. राइट, इसीलिए वो आप किसी भी सेल को ले लो, किसी भी यूकैरियोटिक सेल को ले लो, उसकी सीक्वेंसेस, हिस्टोन के सीक्वेंसेस, द अमाइनो एसिड सीक्वेंस काइंड ऑफ सिमिलर है. क्यों? क्योंकि कहीं पे भी हिस्टोन है इन अ यूकैरियोटिक सेल्स न्यूक्लियस, इट हेल्प्स इन कॉम्पैक्टिंग द डीएनए. ओके, उसका जो फंडामेंटल फंक्शन है सेम है. इज इट फॉलोड एवरीवन? Okay, now coming to the non-histones. Now, no, these non-histones are all proteins which are associated with DNA apart from histones. Okay, whatever proteins are associated with DNA apart from histones are called as non-histones. Okay, now many non-histones are acidic proteins, so they can even have a net negative charge as well. Non-histones include proteins that play a role in the processes of DNA replication, repair, transcription, and recombination. In contrast to the histones, the non-histone proteins differ markedly in number and types from cell type to cell type within an organism. Okay, at different types of the same cell type and from organism to organism. So non-histone proteins, they vary a lot, not only in the different species but also in the cells of the same species, or also in the cells of at different stage of life in the same organism. Okay, whereas histone proteins, they are kind of constant. Now let's talk about the eukaryotic chromosome packaging. So with the electron microscope, different chromatin structure are seen. Okay, with the most relaxed chromatin structure is seen during the interphase, and the most compact structure of the chromosome is seen during the M phase of the cell cycle, that is during the division phase. Okay, now the least compact form is the 10 nanometer chromatin fiber, which is called as the 10 nanometer chromatin fiber, which has a characteristic bead on a string morphology. Okay, and beads have a diameter of around 10 nanometer. Okay, now what is this bead form of? This bead is formed by nucleosome. Okay, this nucleosome has a histone core. All right, this histone core comprises of two subunits each of 2a, H2a, H2b, H3 and H4. Okay, so two subunits each of H2A, H2B, H3 and H4 are present. Okay, so this nucleosome core is actually an octamer. Okay, so it consists of core of 8 histone proteins, that's why it's an octamer, 2 each of H2A, H2B, H3 and H4 around with a, so this is the nucleosome core, right? Now around the core the DNA will be wrapped. Okay, and that DNA, the size of the fragment that is wrapped is around 147 base pair segment of DNA is wound around 1.65 times. Okay, right? So the ball will be there, which is made by, which is called as the which is made by the histone core, that is formed by two subunits each of H2A, H2B, H3, H4. Okay, and around it a 147 base pair long DNA will be wound around 1.65 times. Okay, now this structure of DNA wound around the nucleosome core compacts the DNA by a factor of around 6. Okay, and the individual nucleosomes will be connected, so that is how the DNA will be wrapped around. Okay, the nucleosome core, and individual nucleosomes will be connected by a strand of linker DNA. Okay, so you see here the DNA that comes between the two adjacent nucleosome is called as the linker DNA. Okay, that is the primary packaging. Okay, now, the length of the linker DNA will vary within and among organism. In case of humans, the linker DNA can be of the size of 38. This linker DNA can be of the size of 38 to 53 base pair. Okay, see the segment of DNA that is wound around the nucleosome core is constant, but the size of the linker DNA will vary. Okay, now the next level of chromatin condensation is brought about another histone molecule called as H1. Okay, now this H1 is not a part of the nucleosome core. All right, it will come from outside. Okay, this H1 is a single H1 molecule will bind both to the linker DNA at one end of the nucleosome as well as to the middle of the DNA segment wrapped around the core histones. Okay, so as you can see here, this yellow portion, right, that is H1. All right, so binding of H1 causes the nucleosomal DNA to assume a more regular appearance with a zigzag arrangement of around 30 nanometer in diameter, which is called as 30 nanometer chromatin fiber. Okay, so here with the nucleosome, it was only 10 nanometer. Okay, then it will be arranged, which will have a wide width of 30 nanometer. Okay, so that is called as the 30 nanometer chromatin fiber, which is also considered as the secondary level of packaging. Okay, now in the interphase, okay, when we see the chromatin as the most relaxed, as the thin fiber like thread, okay, that in that time the chromatin exists into this 30 nanometer chromatin structure. okay, in this format only, in this 30 nanometer structure, okay, in the interface. All right. Is it followed, everyone, so far? Quickly, can I have some responses? Okay, now this 30 nanometer is not alone. Okay, this 30 nanometer will further be coiled, right? Because we see that the chromatin structure is the most relaxed when we see thin thread like in the interface. But when it starts, when the cell division starts, the chromosomes keep on getting compact, right? So that compaction how happens, the further packaging. Okay, now this 30 nanometer structure are folded into a series of looped domains which further condense and chromatin fiber into a structure that is 300 nanometer in diameter, okay, which is also called as the solenoid model. Okay, where the chromosomes, this nucleosomes which are arranged in a zigzag pattern will further be coiled, okay, like this. Fine, that will form the solenoid. Now this coiled chromatin fibers are then compacted into a chromosome arm that constitutes a chromatid. Okay, and how it happens, that this 30 nanometer chromatin fibers will attach to some chromosome scaffolds. Okay, and that scaffolds will be provided by the non-histone proteins, you see. Okay, around it, the nucleosome, the 30 nanometer chromatin fiber will be looped like a flower. Okay, so that will do the further compaction. All right, and that is how the DNA will be arranged in a metaphase chromosome, which is supposedly the most compact structure of the chromosome. So in the overall transition from a fully extended DNA helix to the extremely condensed status of the mitotic chromosome, a packing ratio of 500 to 1 must be achieved. Okay, now to give you a better idea, this is the consolidated structure. Okay, so you have a DNA which is double helix, right, bond around each other. Okay, then what will happen, it will be wrapped around the nucleosome core, okay, which is an octamer of histone protein. All right, so this is the nucleosome core, and then again it will be joined by the H1 that will again arrange these nucleosomes in a zigzag pattern, resulting into the 30 nanometer fiber, right? That 30 nanometer fiber will be present in a solenoid form like this, like a circle, flower, okay, with the help of non-histone proteins as well. Okay, and that will result into the ultimate compaction of a chromosomal arm, which is around 70 nanometer diameter. Okay, now after we have discussed about this chromosome compaction, okay, this is the basic chromosomal packaging. All right, however, chromosomes are not rigid structure. Okay, it's not a rigid structure. All right, so rather there will be many regions in the chromosomes. They have dynamic structures that unpack when the genes become active and pack when the gene cease their activity. Okay, and that is one of the way how the gene expression is controlled in the eukaryotic cell. So most dispersed cell of chromosome will be present when the DNA is about to replicate, that is in the S phase, and the most compact, most condensed state of DNA will be present when the cell division is going on. Okay, is it followed, everybody? So क्रोमोसोम ऐसा नहीं होता है कि पूरा क्रोमोसोम और सारी क्रोमोसोम इतनी ही कॉम्पैक्ट स्टेज में प्रेजेंट होंगे. Okay, it's not a rigid structure. So the portion of the chromosomes where the gene that has to be actively transcribed are present. Okay, so that DNA has to be easily accessible by the transcriptional machinery. Okay, तो उन सब areas में क्या होगा, the DNA will be open, it won't be so much compacted. All right, so that the transcriptional machinery can easily reach the DNA. All right, but in those regions where the genes are not present, or the gene that has to be actively transcribed is not present, then the chromosome will be present in this compact structure. Okay, now there are forms of chromatin. So these forms of chromatins are distributed on the basis of the chromosome staining properties. They are divided into two types, euchromatin and heterochromatin. Okay. So euchromatin is the region of chromosome that shows normal cycle of chromosome condensation and decondensation in the cell cycle. Okay, normal cycle means कि वो interphase में relaxed होगा और the division phase में compact होगा. Okay, that it will be relaxed in the interphase and compact in the division phase. This eukaryotic DNA contains the genes which are actively transcribed. Okay, and euchromatin is devoid of repetitive sequence. Okay, so if you see here, you can have a good comparison that euchromatin structure, you see how relaxed it is. The DNA is easily accessible by the transcriptional machinery. Okay, whereas the heterochromatin region is the one which remains condensed throughout the cell cycle. Okay, it does not follow the condensation and decondensation cycle. Okay, this heterochromatin DNA often replicates later than the rest of the DNA in the S phase, and the genes within heterochromatin DNA are usually transcriptionally inactive. Okay, that's why they can afford to be in so much compact structure. All right. Okay. Now, when we see the chromosome, eukaryotic chromosome, there are two very important portions in the eukaryotic chromosome. Okay, that is the centromere. One is centromere, another one is telomere. Okay, so we'll talk about each of them. Now, what is centromere? Centromere is usually considered as the primary constriction of the cell, right? The separation of homologs during mitosis and meiosis depends on the centromere. Okay, now many people often say that centromere is like a glue that holds the both arms together. As you can see here, there are two arms. Okay, now that is absolutely wrong. Centromere is not holding the arm. Centromere is just a part of the same DNA. And as we have already seen that in a chromatid, it is the single DNA that is present from top to end. Right? It is the single DNA present. So it cannot be a glue or anything, centromere. Right? So the centromere is present as a primary constriction. But this centromere is extremely important to make sure that the chromosomes are properly distributed at the time of cell division. Okay. The minimal region of the centromere that supports the function of chromosomal segregation is called as the CEN region. Okay. Now, why this centromere is so important? Because on this centromere, a protein complex called as kinetochore complex assembles. Okay. And it is this kinetochore complex which will attach to the mitotic spindle microtubules coming from the opposite poles. All right. So if there is no centromere, then no kinetochore will attach. So there won't be any attachment site for the spindle fibers. And that's when the chromosome will fail to segregate at the time of cell division. Okay. So acentric chromatin fragment, if it is formed in the cell, it will be lost. It will fail to segregate properly to the daughter cells. Okay. So within this, now this centromeric region is a heterochromatic region. Okay, so that is, it will be consisting of various repetitive sequences and it is usually transcriptionally inactive. Okay, and it is very densely packed. So heterochromatic region of the chromosome, the DNA binds a platform of protein, which is multicellular organism, includes the kinetochore that binds to the spindle fiber during the division. Okay, so that's why these centromeres are extremely important to make sure that the chromosomes are properly distributed. Okay, now coming to the telomere. So where are telomeres present? They are present at the tips of the chromosome, these red regions, right? They are the telomere. So a telomere, a specific set of sequences at the end of the linear chromosome. Okay, what is the function? It stabilizes the chromosome. Okay, and it is required for replication as well. Okay, now see this telomeres, they kind of act as an insulators. Okay, so basically what happens when you have a cell? Okay, there are 46 chromosomes present, right? Now each of these chromosomes, they will have a open end of DNA. right? So it is very possible that two or more chromosomes can join to each other end to end, okay, and that will result in a fusion chromosome, a bigger chromosome. But that doesn't happen in the cell, okay. Each chromosomes, they are maintained as a separate entity, all right. So how that is possible? The reason is because of the telomere, okay. So telomeric region, it kind of provides an insulator, it kinds of provide a cap, okay, so that two different chromosomes cannot fuse with each other. Now, in most organisms, the telomeres are positioned just inside the nuclear envelope and often are found associated with each other as well as with the nuclear envelope. Most telomeric sequences may be divided into two types. So, two types of DNA are sequences are found in the telomere. So, what are these? One is simple telomeric sequences, and another one is telomere associated sequences. Okay. Now, if you see the telomere, so they are at the extreme ends of the chromosomal DNA molecule. Depending on the organism and its stage of life, there are around 100 to 1000 copies of repeats present one after another. Another, okay. So the telomeric sequence will have tandemly repeated DNA, okay. It will have repeated sequences present one after the another. So they are tandemly repeated DNA sequence, okay. In humans and all vertebrates, the repeated sequence is 5 prime to 3 prime TTAGGG. Okay, this is the repeat sequence that you will see in human telomere as well as all other vertebrates also. The telomeric DNA is not double stranded. Okay, so at the telomere, the DNA is not double stranded, but it has a overhang. Okay, and the length of the overhang will vary. Okay. Now, this telomere, just to give you some extra information, so there is an enzyme called as telomerase. Have you people heard of it? Yes, anyone heard of this telomerase? Yes, some responses again. Right, Jason, you have heard of it? So, what does this telomerase do? This telomerase is responsible for the replication of the telomere. Okay, that's why it is called as telomerase. Right now, why a cell requires a new kind of enzyme to replicate? Anyways, DNA polymerase replicates DNA. So why DNA polymerase cannot do here? Okay, because of this overhang situation that is present at the end, DNA polymerase cannot replicate this region. Okay, so if we have to replicate this overhang, then comes the function of an enzyme, which is telomerase, which is essentially a reverse transcriptase enzyme. Okay, so this telomerase is only active in the adult germline cells, in the fetal cells, or in the tumor cells only. Okay, in rest of the somatic cells of the body, telomerase is inactive. Okay, so because telomerase is inactive, so what happens in those cells? DNA polymerase can synthesize only till here. Okay, so every time DNA replication happens, the size of the chromosome keeps on decreasing. All right, and that is also considered as one of the main reason behind aging. Okay, the other type of sequence that is present in the telomere is the telomere associated sequence. They are regions internal to the simple telomeric sequences. These sequences often contain repeated but still complex DNA sequences extending many thousand base pair in the form of chromosome ends. Okay, now we are almost at the end of the class, everybody. So we have unique sequences in the DNA. Okay, we have unique sequences and repetitive sequences. Okay, so what are unique sequences? So unique sequences, they will be present only one to a few copies in the genome. Okay, so usually the unique sequences will be present only one time in a haploid set. So if you are talking about a diploid cell, so it will be present in two copies. Okay, there are moderately repetitive sequence which are present in few to about a few copies in the genome. In the highly repetitive DNA sequence, that is present around 1,000 copies. Sorry, it's missing 1,000 copies in the genome. Okay, now in prokaryotes, all the genome is present as a unique sequence. Okay, most of the DNA is present in the prokaryotic genome as unique sequenced DNA. Okay, except the rRNA and the tRNA gene. The only exception here is rRNA. This is exception, okay, not example. Only exception are the rRNA and the tRNA genes. Okay, they are present in many copies in the prokaryotic genome, but apart from that, entire DNA is unique sequence DNA. Okay, it doesn't have any repetitive sequences. Whereas eukaryotic genome, okay, it consists of both unique sequence and repeat sequence. So usually the unique sequences will be the protein coding genes, right? But apart from that, there will be a various amount of repeat sequences as well in the eukaryotic genome. And there are various types of repetitive sequences that are present in the eukaryotic genome. All right, so here we come to the end of the session. So definitely we are going to discuss some questions. So come on, give me some thumbs up, give me some BIOs. Are you all ready to see some questions now? All right, so I have taken some of the previous year questions. So RNA is genetic material in some viruses and prokaryotes, virus, prokaryote, primitive eukaryotes, some viruses only, none of the above. So as you have seen in the session, it is only some viruses. Next, nucleotide base pairs usually present in nucleosome are 147. Okay, this fact-based questions can be asked. All right, and also how many turns it takes. So it will be 1.65. So this fact-based questions you should remember. Telomere repeat sequence in humans. Okay, that question we have seen, right? TTAGG, that is the answer. That is the telomere sequence in humans, and not only in humans, but also in the vertebrates. SARS-CoV-2 is caused by a single-stranded RNA virus. Okay, so definitely you can expect a question from SARS-CoV-2, anything related to coronavirus. Okay. Nucleosome core possessed with what is nucleosome core made up of? Two subunits each of H2A, H2B, H3 and H4. Answer is option C. Protein responsible for the structural maintenance of chromosomes are. Okay, now see, this part we haven't seen, but I'm going to tell you now that there are two non-histone proteins that are present on the chromosome. Okay, by name we'll discuss. Condensine and cohesin. Okay, now this cohesin is present, it holds the two sister chromatids together till they separate in the anaphase. Okay, so cohesin holds sister chromatids together. Okay, whereas condensine is the non-histone protein that helps in the chromosomal assembly. Okay, so it maintains the compaction, or it causes the compaction of chromosome or the chromatin fiber. Okay, so the protein responsible for structural maintenance of chromosome will be, answer will be condensine. Which of the following is not true for euchromatin? They are transcriptionally active. Yes, it is true. They are less condensed region of the chromosome. Correct. Bar body is an example of euchromatin. That is something which is wrong. Bar body is what? It is the inactive portion of the chromosome that happens in the that occurs in the female cell because of the dosage compensation for the X chromosome, the extra X chromosome that is present. Right now, this bar body is the heterochromatin. Okay, it is made the portion of the chromosome is made compact so that it becomes transcriptionally inactive. Okay, that is what is called as bar body, and bar body is an example of heterochromatin. So that's why this is untrue. Okay, not true. DNA gyrase is required for. So as I told you, DNA gyrase is a type of topoisomerase 2. So topoisomerase means it will cause supercoiling, right? So DNA gyrase is required for supercoiling. If it talks about the type of organism, then you have to remember that it occurs in the prokaryotes only, bacteria only. The enzyme responsible for removal of DNA supercoils. Okay, so it will be topoisomerase. Next, histones are rich in, so histones are proteins which are made up of lysine and arginine, the basic amino acids. Vishal, I did not get it. Sorry. Which question are you talking about? About this first question, protein responsible for structural maintenance of chromosome? So in that question, A only is correct. Condensine is the correct answer, which maintains the structure of the chromosome. Cohesin is only required for sticking the sister chromatids. Euchromatin, yeah. So about the euchromatin, yes, it is transcriptionally active. So it is true only. You have to select the not true. Euchromatin is transcriptionally active region. Heterochromatin is the transcriptionally inactive region. So you have to select the false statement, isn't it? All right. So with this, we come to the end of this session. Optimism is the faith that leads to achievement. Nothing can be done without hope and confidence, as Helen Keller said. So you have your hope. BioTecNika is going to build that confidence in you that you will be able to clear your PG entrance exam. Helicase, Shrivani, helicase unwinds the strands of the DNA. In DNA replication, the double-stranded molecule has to become single-stranded, right? So helicase helps in removing the helical turns. All right. Thank you everyone for joining. Here is again the information about our BioTecNika app, which you can download from Google Play and Apple Store, and the BioTecNika Telegram group that you can join for more classes, podcasts, webinar courses, newsletters, job vacancies, and many, many more things. I'm very sure this session was helpful to you all, and you guys will keep joining our sessions from the Prarambh series for more discussion on the different topics, different units of the syllabus. Please don't forget to fill up the feedback link provided to you at the end, and with that, you will get the notes of the session. Thank you, everyone. Bye. Good night.